So now we talk about Lender-Waterman curve. Lender-Waterman curve or equation is an important equation that is used in genome assembly projects. It correlates the coverage versus the size of the context. So it's an interesting and important subject to talk about. We'll talk about this topic into these following sections. So what they suggested, Lander and Waterman, was that this distribution or the of these reads or the number of times a base is sequenced follows a Poisson distribution. We'll talk about different distributions in our stats review kind of a lecture. They made two key assumptions. Number one was that reads are randomly distributed in the genome, so there is no biasness, so reads they can come from anywhere in the genome. And number two is the ability to detect an overlap between two truly overlapping reads does not vary from clone to clone, so there is no effect of the clones actually on these reads. What the model is about? So model says that if an overlap length was larger than certain cutoff or threshold T, then the reads should be merged into a context. So we keep on merging the reads into the contexts as per this model, and if their overlaps are greater than some threshold, unless we keep on doing until no more reads are merged. So here are different variables which we use for this equation, where G is the haploid genome length in base pairs. Haploids, as I said earlier, is the one set of chromosomes. So for example, in human, there are 46 chromosomes, but actually there are in two sets of 23 each. L is in this equation sequence read length, N is the number of reads, and T is the amount of overlap needed for detection in base pair. So that thing is T. Coverage, we have already seen that. So coverage is defined as basically the how many times a base is sequenced. So if we take the whole genome, so it can be given by this equation. So where L is the length of the read, N is the total number of the reads produced, and divided by the genome size, haploid genome size in base pairs. So we can get the gap length from these two equations as per this Lander-Waterman model. Let EC, e raised to power negative c, be the probability that the base is not sequenced. So it's the probability that the base is not sequenced. We can get the total gap length as it's equal to the percentage of the genome not sequenced, this thing, into total genome length in base pairs. So it's e raised to power minus c into g. And the second equation is how can we get the total number of gaps? So obviously it's percentage of genome not sequenced, which is e raised to power c, into the number of reads, which is n. So these two equations are what we call them as Lander-Waterman equations, and we believe they are important in those genome sequencing and assembly projects. So we can calculate the sequencing reads needed to cover the genome, and then we can also have an estimate of resulting contexts. So for example, in other way around, if we want to know about how many contexts we can assemble, we can estimate it through the coverage. So in this equation, you can correlate coverage versus number of contexts, as we see here. So number of contexts is on y-axis, whereas x-axis is your coverage. So we see that if the coverage is really low, like 1 or something, so you will have a huge number of contexts up to over 3000 contexts, which is actually there are small size contexts. As you move towards the right and you increase the coverage, number of contexts start decreasing, as we see here. So we have roughly like these 500 contexts per 5x coverage. So number of contexts, as we increase the coverage, they decrease. So what we need in our good assembled genome, do we need long contexts or do we need short contexts? So what do you think? So good assemblies are the ones where we have the context size are larger. Obviously the shorter contexts, they are kind of giving you an idea that assemblies might not be as good. So far what we have seen here is that this Lander-Waterman equation is an important equation, and that is important to estimate the coverage versus number of contacts. So it can be used to estimate a good assembly and how much coverage is needed in order to get a good assembly before we start our experiment.